All right, so we're going to start with obviously bone. And then kind of just want to break down through the two divisions of bone. So we have two major divisions. What are the two divisions that we break the skeletal system into?
Axial and appendicular.
Perfect. And then axial is what makes up the axial? You can just
I thought my mic was on. That's going to be your skull, your vertebrae, and your auditory ossicles, as well as any bones within that area.
Perfect. And then appendicular, I always just say is the appendages, so everything outside of what's like central to the body. So arms, legs, things like that. I do know he really loves to go through the types, like specifically the classifications. He likes asking questions on these. So we'll just go through these really quick. Oh yeah, UTA is here. She's here to answer any questions you guys have in the chat. So I won't have a lot of time to look at the chat. If there's something pressing you need to ask, just unmute and ask. But if there's something you can just throw in the chat, she's there to help out. Okay, so what are some examples of long bones in the body?
I don't know how long we'll be able to last. Yeah, I don't know, 2 hours. Till 9 o'clock. It's definitely not.
Your femur, radius, ulna, humerus.
Perfect. I'm going to also just put appendages just to save some time, but basically things that make up our legs and arms, right? So all those longer bones, that includes our fingers, so our phalanges, our metacarpals, things like that. What's like the major function for long bones?
Support and structure.
Yep, support and structure. They also allow
Movement.
us to move, right? Muscle is usually attached to these, so it allows for some movement. All right, what about our short bones?
Carpals and tarsals.
Perfect. Oops. Yeah, that's way bigger now. So carpals from the wrist. We have tarsals. What's their major function?
It's your mom. Kitty, kitty, kitty.
You guys have any sort of idea for function of short bones?
To allow for movement, but restrict that movement to keep the joint stable.
Yep. So movement. I'll probably just write movement for now, but knowing those details are good. What about some flat bones?
I know the skull.
Yeah, the skull. This seems very tedious, but knowing these is important. So the skull, I always also add like the ribs are obviously pretty flat, sternum, things like that. And these are usually used for protection. So usually they're surrounding, obviously we know our rib cage is surrounding our lungs and our heart. So that's surrounding those vital organs. What about irregular bones?
Isn't it like the spinal cord?
The spinal
and
cord,
like
yeah.
Bones in the face?
Yeah, so be careful when we say spinal cord, right? Because cord would be nervous tissue, but we could say vertebral column. So all those vertebrae that line that protect the spinal cord for sure. That all sits in there. Say, no, you're fine.
Do the coxals count as irregular or flat bones?
They'd be irregular. And I'm pretty sure the hip, so like the pelvis, because it hasn't obviously. I like to just think of the irregular bones are the ones that don't have they have a very odd looking shape to them. Typically, these are for muscle attachment, or also we could say support. Obviously, when we think of our vertebra, that supports most of our body weight and holds us to the positions that we can move in. So we like to think about support for that one. And then what about the sesamoid bones? I think there's really only one.
That's gonna be your patella.
Yeah. So patella basically protects. I think there's one more, but I'm just going to focus on patella. Protects the joint, specifically the knee joint, right? Sits in front of the knee. So again, know these, feel comfortable with these. He'll ask questions about them. Oh, and then the last one, sutural or Warman bones. Really only one place.
Doesn't it like connect like the skull? It gets like little tiny bones in between the seams.
Yeah, and like if you've taken anatomy or if you've seen a skull, they're the sutures, so they're like the lines that almost line those flat bones that make up the skull. So I'll just say probably protection, support, and then provide like connection. I meant to add, my spelling can sometimes be really awful, so don't go based off of that. Cool. Questions on any of that stuff? I know that is very basic stuff, but if not, we'll go to gross anatomy of the bone. So I left something blank. These notes are posted on Canvas under the exam info page. You guys can find those there. Okay, so let's just start with really the bone. We don't need to do all the extra stuff yet. So looking at this right here, what do we call the ends of the bones, or long bones specifically?
Epiphysis.
Yep. Epiphysis. And usually we have two ends of the long bone. And then the way that we name them is dependent on their proximity to the joint. So this is a femur. So we would say the proximal epiphysis is at the top one because it's closer to the actual hip joint. And then the other one is the distal because it's closer
Your
to the
distal.
Yep. I added metaphys. I wouldn't worry too much about that one. Kind of the big ones are going to be epiphyses. And then what's the middle section called?
Diaphysis.
Yep. Cool. So very general, that's the structure and the anatomy of the long bone. Now we'll go into more of what each compartment is kind of made of. So what lines the two ends or the epiphyses of long bones?
Hyaline cartilage.
Perfect. Yep. So it's a type of, should add that actually. Hyaline cartilage. What do we call that cartilage specifically?
Articular.
Yeah. And I like to think of it as it's articulating with the joint. So usually these are lining places where there's a joint, and it's to protect that bone from rubbing on bone. So that hyaline cartilage is the type of cartilage that it is, but we call it articular because it's articulating with the joint. And you end up having a synovial capsule and all these things that encapsulate that actual joint. But knowing that it's hyaline is a good thing. What cells make hyaline cartilage?
Chondrocytes?
Perfect. Yep. Chondrocytes. So we know chondrocytes make all our types of cartilage. The type of cartilage that it is dependent on how much or what's in that actual extracellular matrix. So for elastic cartilage, if there's a lot of elastin there, then it's going to be elastic. All right, so moving down our bone, we have our spongy bone, which is also trabecular bone. This is what covers the two ends, the epiphyses. What is located inside of the spongy bone?
The red bone marrow.
Yep. So red bone marrow, this is where we have our blood cells being made, our immune,
cells.
Yep, white blood cells, erythrocytes, all of that is being produced here in our bone. I went ahead and added a epiphyseal line. We'll talk about a little bit about that when we talk about bone growth and long bone growth. But usually the epiphyseal line is made up of chondrocytes before puberty or during puberty. But after puberty and once we're all grown, these lines will be filled with calcified bone. So then you end up with almost like a line inside the bone that you can see where that cartilage died, and then the bone replaced it. From here, we have our endosteum. That's considered, I tried to draw it well, but it's that inside lining. So the inside lining of the bone, or where the compact bone ends and you get into the medullary cavity is called your endosteum. The outside layer is called our periosteum. And then this guy is going to be our compact bone. So we're made up of compact bone. What is compact bone made up of? What's like the big component of compact bone? So
Osteons.
osteons, yes, that is like the functional. It's a great point. So it's made up of osteons, which are like our functional unit of compact bone. What's like the specific makeup of compact bone that actually makes it hard and more less frail, I should say. Not frail, that's a bad word. More dense. There's a specific
Hydroxyapatite.
yeah. Hydroxy crystals. So these are going to be basically calcium phosphate crystals that make that compact bone super compact and dense and allowing us to have some sort of compression to our bones. So it's really about that protection side of it. And we'll get into osteons, I promise. But yes, osteons make up compact bone. They're the functional or structural unit of compact bone. All right. So inside this cavity, we call it our medullary cavity. What do we have actually inside the medullary cavity?
Adipose
marrow.
tissue fat?
Yeah. And we call this yellow bone marrow, which kind of gives it the yellowy color. It can be adipose. So basically fat storage is what's resting in that medullary cavity. Cool. So again, knowing the epiphyses, diaphysis, knowing what's located where is going to be pretty important for this exam. Do you guys have any questions on this gross anatomy stuff for bone? Cool. If not, I did not have any blanks on this because I'm just going to kind of talk through it because it's kind of a complicated picture. But this is going to be what actually makes up that compact bone. So now we're jumping back to those osteons. So looking at compact bone, we end up having a bunch of these osteons that circle around each other here. And as they get more compact, we end up with osteons kind of stacking around, creating that nice compact shape for that bone. On the inside here, I added endosteum because that's this inner lining right here. I have an orange. And then you're going to have some kind of spongy and trabecular bone on the inside there. On the outside, we have our periosteum. So that's over here. So that's lining the outside of the bone. And then if we really took a closer look at these osteons, I think this picture is a little bit more important because it has exactly what we kind of need to know. So within an osteon, we have these lamella, whatever you want to call them. So there's these concentric rings that are formed due to what cell type makes the hydroxyapatite?
Osteoblasts.
Yep. So osteoblasts are going to start building those lamina. I can never say this word. Lamellae. Anyway, they build them outwards. So these are growing, causing these rings. And within these rings, we end up with what we call lacunae. So this here. And lacunae are essentially a space. I'll just say space. And it's a space that houses osteocytes. And knowing just from kind of how when we talked about bone formation, as osteoblasts build that matrix and it's coming outward, it slowly starts to actually surround osteoblasts, which then they turn to osteocytes. So once they turn to a site, they no longer make that hydroxyapatite. They're actually just maintaining the bone. So they're the ones that are doing all that rank L, SOST, OPG, all of that stuff is coming from those sites. because now they're just maintaining it. They're getting signals. They're telling the blast in the site or class what to do. So lacuna are a space that these osteocytes live in. From there, they'll have little projections called caniculi. This allows for these cells to communicate with each other. So that's where we can bring in our idea that we talked about in the first unit about gap junctions, things like that. This is where these cells can actually have some sort of cytoplasmic continuity, and they can have, you know, share ions, share signals, whatever it is. And then basically just the center of it is that central canal. You'll have some sort of blood supply there, vein, artery, you'll have a nerve, and all of that will run kind of in the middle of these osteons. So then if we're coming back up to this picture, you can see all of them have somewhat of a central canal that gives them a blood supply and allows for, you know, signaling and things like that. You guys have any questions on osteons? What, just while we're at it, what cell type do we find in the endosteum? So out of our bone cells that we learned, which we only really know 3 of them, which ones live in the endosteum? Jeez, I can't talk.
Is that going to be the osteoblast because they're building?
So these will actually be our clasts.
Classed, okay.
Yep. So clasts are going to live in the endosteum, and they actually break down the trabecular bone. So when they're actually reabsorbing bone, they're using that trabecular side. They're not really breaking down. I mean, they could be breaking down a little bit of the compact on that inside, on the inside of that endosteum, but they're going to be there, while our blasts are the ones that are in the right under the periosteum. So I should say they're like in this layer right here. Just a little detail to have, just so you kind of have some orientation. And obviously our sites are living in here in the lacunae inside those osteons. Cool. Questions on osteon stuff? If not, sorry, I feel like I'm going kind of fast, but I feel like endocrine and nervous system are a lot more complicated. So I'll try to get through bone a little bit faster. So really quickly, we kind of already talked about it, but let's go through our bone cells. So I have 2 pathways here. So kind of where they originate is kind of the most important thing for me. So coming from osteogenic cells, or we could say osteogenic stem cells, meaning they can differentiate into different things. What is the cell type, and I wrote it here too, that lives in the periosteum?
Is it the osteoblast?
Yep. These are osteoblasts. And then what do osteoblasts become once they're locked into the lacuna?
Osteocytes.
Perfect. So once they're locked in the lacuna, they become the sites. They can now maintain bone using rank, SOST, all that fun chemical stuff. And then coming from kind of a completely different line is going to be from our monocytes or macrophages. These are part of the immune system. These can be differentiated into what cell type?
Osteoclast.
Perfect. So we know clasts are the ones that are doing all the bone absorbing or reabsorbing. So they'll be breaking down hydroxyapatite using hydrogen ion. And I promise we'll go through this system because I think the rank system is a good system for him to ask questions on for sure. Cool. Any questions so far? If not, I'm going to move on to our very fast breakdown of bone formation, and then we'll go through bone remodeling, and then that's kind of it for skeletal. All right, so these are the 2 types that bone will form. So these are all happening in fetal tissue. So within the womb and the child's growing, these are the way the bones are actually being formed. So we have 2 types. We have intramembranous ossification and endochondrial. Chondrial, I feel like, is a little bit more important because that's where our long bones, that's where we get a lot of the cartilage stuff. But I think intermembranous is also important. So I'm just going to go through like quick steps of these. So the first step for intramembranous is we always are going to kind of start with mesenchymal cells. And we should kind of remember mesenchymal comes from, these are the ones that make connective tissue, and we know that bone is a type of connective tissue. So all connective tissue comes from mesenchymal cells. They're a type of stem cell. They differentiate into whatever type of tissue you're looking for. But because we're talking about bone, these mesenchymal cells are going to start off by becoming osteoblasts. And this is all kind of happening inside of almost like a pool of cells, like a little matrix of cells. So we're not starting with cartilage like we do with endochondrial. We're having like a little pool of just mesenchymal cells that then differentiate into osteoblasts. From here, the osteoblasts congregate to ossification centers. So they're going to kind of come together into these centers where we're going to actually start creating bone. At that point, they start secreting osteoid, which is basically just the stuff that makes up hydroxyapatite or compact bone. So we'll say secrete osteoid and calcium, which will, create compact bone. And like I said earlier, once that calcified bone traps osteoblasts, they become osteocytes when they get trapped in there. And then due to the fact that mesenchymal cells, like I said, they're part of making connective tissue, blood and things like that are essentially a type of connective tissue. So some of them will start to actually differentiate into capillaries and making up blood vessels. So once there's a blood supply present, osteoblasts make that trabecular bone. And I kind of actually, there's like an image I think he had on his slides actually. It showed like a capillary was moving kind of through all the projections from a trabecular bone. Just thinking about those osteoblasts are creating that matrix just around all the capillaries, which makes it more spongy looking and less dense because it's moving around. It's not getting as compact. And then really our last one is the surface is making that compact bone. So this way is really starting off with this weird pool of cells that then start to differentiate into blasts. We start making the matrix and things start forming from that little intramembranous pool. Now for our osteochondral or endochondrial, we start with same ones. Yes. There are questions, sorry. Oh yeah, I meant to add. Flat bones are mostly intramembranous. Long bones are going to be typically your yeah, long bones will typically be your endochondrial. All right, so starting here, we still start with mesenchymal. What do you think these turn into in this case instead of the other case? So when we're talking about intramembranous, they turn into osteoblasts, but what about endochondrial?
Chondrocytes.
Yeah,
Which we already kind of talked about make up cartilage. So number 2 here is chondrocytes form that matrix of hyaline cartilage. So they form hyaline. Once that hyaline cartilage is formed, you are going to have some osteoblasts start creating, start to calcify. calcify matrix, which will end in cell death of chondrocytes. And really that's due, I should have added this first actually. Let me just throw it right here. It's due to blood supply is what brings those osteogenic stem cells, the ones that we talked about that differentiate into osteoblasts. So when blood supply occurs, that's when those bloods or those bone cells can get to that hyaline cartilage and start making that matrix. And then from there, we kind of basically will have some bone development. And that's starting kind of in the center. So I'm going to draw a picture of this just so we have. So here's my hyaline cartilage. From here, once we get that blood supply in here, here's my capillary. Bone will start to form, but really it's starting to form from the middle of the bone. So we'll have compact bone forming here. And then really we start growing out that way. That's what starts pushing the cartilage farther away. So then we end up with those epiphyseal lines like we were talking about earlier. So during puberty, your bones are still growing. And obviously once you're born, your bones are still growing because you're a little infant and then you start getting bigger from there. So those chondrocytes keep getting replaced by osteoblasts and osteocytes, making that bone grow longer. And then eventually those epiphyseal lines or that last little bit of chondrocytes will end up dying, giving you that epiphyseal line once your bones are completely grown. Any questions on that? I think that's kind of some of the most complicated stuff for this unit is the formation. I know in his slides I just don't have time to go through them in depth because I'd rather get to endocrine and nervous system. But if you feel like you're questioning yourself a little bit, go look at the slides that go through each center of ossification. I think there's like 4 slides that show different ossification centers and what happens in each one. Just knowing those briefly, I don't think they need to be anything in detail, but if I had more time, I'd go through it. Questions on this though, before I move on to the last part of bone? Cool.
I have a question.
Yeah, what's up?
What does it sort of look like when the osteoblasts are congregating in intramembranous ossification? Are they like kind of clumps floating around?
Yeah, so it's kind of, I'll try to draw. So usually it starts like a pool. And we start with mesenchymal cells. So these cells are just kind of chilling out here. And once they differentiate, they'll congregate to like a center that we call the ossification center. And then they'll start building bone. But remember, they stay on the outside. So as this bone is growing this way, the blasts, so like let's say my bone is out here now, our blasts are staying out here. And they're growing it out that way still. Did that kind of help a little bit?
Yeah, that's really cool that they can form the right shape like that.
Yeah, it's pretty crazy. I think that's why, and it makes, for me, it makes a lot of sense that intramembranous does the flat bones because they just have more of a unique, like they can grow out that direction and it can be a different shape. When you look at the endochondrial, with them starting to grow right in the middle of that slight bone looking hyaline cartilage, it makes sense that they push it away and make it longer. So I think you can look at it that way too, if that helps, just kind of memorizing what type does what and how it starts really. The body's crazy, y'all. Does some crazy shit. Okay. What else? Any other questions on that? If not, let's talk about bone remodeling, and then this is the last thing for bone. So, starting with bone remodeling, we have our bone. let's say I just like to use the parathyroid hormone example. It kind of ties in endocrine. So the parathyroid gland sits next to the thyroid, or like on top of the thyroid, and it detects the blood calcium levels. So it's looking at how much calcium is actually within our blood at that time. And if our calcium is low, then it's going to want to cause the activation of osteoclasts, because that's what's breaking down calcium to get that back into our blood. And obviously, calcium is a really important ion for almost all body processes. You'll realize in the heart, it runs a huge function there, and just in muscle contraction in general. So let's say we have low blood calcium. So we need to start breaking down some bone. Our first big guy that's released is going to be our rank L. Stands for rank ligand. Ligand meaning a chemical messenger. We refer to a lot of things as ligands. So like neurotransmitters are considered a ligand, things like that. They essentially will send a signal to a cell. So osteocytes, remember, maintain bone. So they're the ones that are really doing this stuff. This is going to come on over, and it's going to talk to a pre-osteoclast. So not a class that's activated. This class is just chilling. It's kind of why we call it a pre-osteoclast, and it's going to bind to a rank receptor. Once it binds there, it's going to tell this cell, all right, we can become an active osteoblast or osteoclast. And now we can actually start breaking down bone. What do the class use to break down that hydroxyapatite?
Acid?
Yeah. Sorry, I was just right, hydrogen ion. But yep, they're going to use acid. So they use acid to break down that calcium phosphate or that hydroxyapatite. I honestly don't really know the formula for the phosphate part, but I just make it up every time. He's not going to ask you that, so don't stress about it. But this breaks up, and now we can then increase our calcium levels in the blood, because now we can release it from the bone, give it to the blood supply. Our parathyroid will be happy, and then we can stop using parathyroid hormone. Now what if we're like, okay, never mind, or there's some sort of issue going on where we don't want bone breakdown? What's the other big signaler that we can use that osteocytes use?
Is it OPG?
Yep. So OPG acts as an inhibitor. And what it will do is it will actually bind rank L so that rank L can't bind to the receptor. So this would actually come on over here. I'll draw this again. Here's my OPG. And if this binds in here, then now it can't go and bind to that rank receptor that's on that pre-osteoclast, basically inhibiting the system. So I'm going to draw a nice big inhibition arrow just so we know that it's inhibiting osteoclasts. So we have no osteoclast activity. So this is actually promoting bone formation. And then we actually have one more that promotes bone reabsorption, also comes from osteocytes. What's that last one that promotes bone? Yep, SSOT. I honestly don't know what it stands for. But SSOT is also going to play a part on osteoclast activity. So it's going to increase that activity of that osteoclast, causing more bone breakdown. Do you guys have any questions on that? I think that pretty much sums up bone as quickly as I possibly could in 40 minutes. Cool. If not, I'm going to move on to endocrine. Do you guys have any last minute bone questions at all? Right, endocrine it is. So let's start with just types of cell communication. I know this is very, very, very basic. So paracrine.
Wait, does the
Go
rank L, does the OPG inhibit the pre-osteoclast or the actual osteoclast?
I think it actually is the pre.
Okay.
Yeah, because I guess if you think about it, rank L, if you're blocking rank L, then you're not going to be able to have it go to the pre-osteoclast. You're blocking it from actually farther above the chain. SOST is the one that's inhibit or going directly to the activated osteoclast that's already active and causing more bone reabsorption. Thanks for catching that. Sorry. And yeah, so hydrogen is released from the osteoclast and it causes the breakdown of hydroxyapatite. Hydroxyapatite is made up of calcium. So in the end all be all, we're actually increasing blood calcium because we're taking the calcium from bone and putting it into the blood. If that answers your question, Kaylee. Cool. I'm going to move on. Sorry, a lot to go through. Okay, so types of cell communication. What is paracrine?
Is it localized communication in the same tissue?
Yeah, exactly. I always think cell to neighboring cell. And yeah, we could say same tissue. I'm not going to use the example I usually do because I don't want to confuse you guys with an exam coming up. But they use this a lot. A lot of cells will use this to communicate to each other, even if it's a cell that's right next to it. So it can like, tell it if there's an infection coming, things like that. What about autocrine?
Isn't that where it communicates with itself?
Yep. So cell to self. And then endocrine is what?
Long distance?
Yep, long distance, usually.
Bloodstream.
Yep, bloodstream. So these things need to get into the bloodstream to get around the body, get to whatever organ or target organ we need to get to. So it's going to go pretty far. And then what about our last one, exocrine?
Direct.
Yep. So this is direct to surface. So usually, I guess we have some endocrine gland or exocrine glands that will be still within an organ, but usually it's to surface epithelium. But there's some in the pancreas that are actually considered exocrine glands that will just secrete stuff into the actual I don't even know what they call it, tubule of the pancreas. I made that up, but they secrete it into basically to get into the intestines and then debuff some of the acid that comes from the stomach. That's considered exocrine, even though it's not actually being expelled out the body. Obviously, our major ones are like sweat, milk, sebaceous, things like that. We'll go through these right now. So merocrine is what type of exocrine gland?
Is that the one where it's pinched off? Like
Yeah,
a piece?
yeah, and I think a lot of it is this one is actually just going to be things are just released and secreted directly. So this will be like saliva. With saliva, we don't actually lose a piece of the cell. It's things are actually just leaving that exocrine gland. So let's just do examples. But this is saliva or salivary glands. This could also be our sweat glands, but not the ones that produce too much odor. Usually the odorous ones are the oil ones. Those are going to be ones that actually chunk off pieces of cell. So then apocrine is going to be the one that you were saying is going to be actual parts of the cell are pinched off. And then this one's going to be our odor glands, or more like our odor sweat glands, the ones that actually stink, or our mammary. And then the last one is the whole cell. Bursts, basically. Ruptures. This would be sebaceous. So oil glands. And really, I think it's just knowing these. So I wouldn't stress too much about how they work because we didn't talk about how they like fully work besides saying that the whole cell ruptures. But just kind of knowing what falls under these categories is important. And obviously we're talking about endocrine mostly, so this is going to be the big topic. But know your paracrine, your autocrine, and your exocrine. Questions on any of that? Cool. So classifications of hormones, this stuff is super, super important. We could literally ask you every unit starting from now about other hormones that you talk about. Because once you get to digestion, we'll talk about insulin, which is a hormone, and what does that do? What type of hormone would it be classified as if it's using this or that? I like to classify them as hydrophilic and hydrophobic because I think that kind of helps with understanding the types of receptors that they're going to be using. So starting off with hydrophilic, what type of receptors do hydrophilic hormones use? So think about just the general idea of hydrophilic. What does hydrophilic mean?
Loves water. So wouldn't that mean that there are wait, yeah, don't mind the cat in the back. Intracellular receptors because they need a secondary messenger and can't just go through the membrane. Sorry, my mind's at a million places right now.
You're good. Because it's funny, you said intracellular, but then you described the correct term, which is great.
Dang.
Intracellular would mean something that needs to go into the cell, which would
Oh,
be
okay. So the transmembrane to let it in.
It's funny because you
Like I said, a
defined
million
it.
different places.
You're good. You defined it perfectly. It was just the first part. No, that's great. So it's going to use, and I just really classify it as a GPCR because that's typically the one that we're using. Okay. But we should probably also just add transmembrane because it could bind to any other type of transmembrane protein also.
Oh my God, wait, that makes so much sense now that I'm actually thinking. Thank you.
Yeah, of course. So GPCRs are usually that big one that's going to cause that secondary messenger. And I promise we'll go through those because GPCRs are like very important for physiology in general. But hydrophilic love water. So they're totally fine with chilling out inside the extracellular space, which is why they can use a transmembrane protein. So what do you think are some of those big categories of hormones that are considered hydrophilic or would need a GPCR?
Amino acid derivatives and peptide hormones.
Yep. And I know you don't have to know the amino acid, like the actual amino acid derivative ones. I think tyrosine is one, and there might be a tryptophan one. Yeah.
That's what he told us.
Yeah, so I think knowing those two would be fine, but I don't think he's going to ask too much detail about like what does the tryptophan actually turn into. Like, don't worry about that stuff. But I also like to add when it comes to these kind of hormones, they are usually also really, really big, which is why they also can't just go right through the cell. So if that helps kind of understanding it, usually if they're amino acids, they're pretty large. And if they're peptide, which is the other one, we know that peptides are chains of amino acids. So they're probably pretty large. They're not going to be able to just burst right through the cell membrane, which is why they need some sort of transmembrane protein. Cool. Let's switch on over to hydrophobic. So what are the classes for, or what, let's see, I'm losing my mind too. What receptor do hydrophobic amino acid or hormones need? See, now I'm stuck on amino acids.
These would be the intracellular ones.
Yep, intra. And they usually act as, I'm just going to put it in parentheses, but they actually have somewhat of an effect on gene expression. So instead of utilizing some sort of secondary messenger where we can increase channel activity or something like that, this one will actually cause genes to be expressed. Either that's making a protein that we're looking for or something like that. What are there's really just like one big class of these.
Lipid derivatives.
Yeah,
lipid.
let's do lipid derivative. I guess there's two. Yeah, lipid derivatives. And then the other one is cholesterol derivatives. And then these are really the big one we talk about a lot is the steroid hormones. So these are things like aldosterone, obviously testosterone, other androgens. So these are the ones that can pop right through the cell membrane and go act directly on the genes themselves. I think there's one other one. Oh, it's part of the lipid ones. It's the ESCO. Eicosanoids, sanoids. Cool. So we can easily ask you, like, this hormone is a peptide hormone. What type of receptor would it bind to? I think that question's way too easy, but we could ask you stuff like that. So knowing kind of their properties, or we could say an unknown hydrophilic hormone would bind to this and cause what sort of secondary messenger? You guys have any questions on that?
Yeah,
What's up?
can you give me a second? Sorry, I'm like, sorry,
You're good.
sorry. It's stupid. I have to make a stupid meme, and I need to know if this is like too stupid and unfunny to post. It's not very funny, but I'm like,
That was not a question for me. I've realized as I sat there and waited. Okay. Let's go to stimulation of hormones. We'll probably hit this pretty quick, but we have three types. Yes, catecholamines are part of that group. So what are the three stimulations for hormones? Basically, when I say stimulation of hormones, just thinking what will cause the hormone to be stimulated or cause it to be released. We just have three like broad categories of this.
Humoral, hormonal, and neural.
Perfect. What is humoral?
That's going to be a change in the extracellular matrix. The way I think of it is way back when the Black Death was like still a thing. Doctors label different fluids and areas of the body as humors. So, okay, if there's a change in fluid, there's a change in the humors.
Yep.
Historians, please check me on that one because I'm not becoming a dietitian to study history, okay?
No, you're good. No, this is actually, I think one of the big ones I like to use is the also I hate history. I actually had an interview, a PA school interview last week. They asked me why I had a C in history. I was like, because it's history. Like, why would I be applying to PA school? If I loved history, I would have been a history buff. Sorry, that was so beyond the point. But so for humoral, it's all about extracellular changes. So instead of it being a hormone talking to a hormone, I like to use this example of aldosterone, which is one you've talked about. And aldosterone is a steroid hormone, so it's going to use an intracellular receptor. It's going to cause an actual change in how many channels we make. So it will make more ENaCs is what they're called. They're epithelial sodium channels, and they'll put them into the wall of the nephron, which is in the kidney, that helps to reabsorb sodium. So this hormone isn't causing another hormone to be released, but it's actually causing sodium reabsorption, which we could say is an example of it's changing the extracellular matrix. It's not causing more hormones to be released. It's really just changing where ions are moving. The other one was hormonal, which makes way more sense, right? It's hormone talking to hormone. And we'll go through literally all of these, but I'll just do this one. But GnRH, which is gonadotropin releasing hormone, causes the release of FSH and LH. And those two, you could think of it as the two hormone, the GnRH is causing another hormone to be released. So that's what's causing the stimulation. And then what was the last one?
Neural.
Neural, yep. And this is usually just neurotransmitter derived. So neurotransmitters chatting to each other, which obviously we talked a lot about in neuro, which we'll try to get to as much as we can. Questions on any of that stuff? Like I said, I think just know these. Like a question could come up about a hormone changes the extracellular matrix. Knowing that that's a humoral response is kind of just a good thing to know.
So humoral could be anything in the blood, whereas hormonal would have to be the hormone actually binding on the cell, triggering that cell to release more hormones.
Yeah, I think that it would have to be hormone to hormone. So like in GnRH is a good example. So that's released from the hypothalamus. That goes through the hypopituitary axis, whatever that vein is, portal system, runs through that. And once it gets to the anterior pituitary, that GnRH will bind to a cell there called a troph cell. And then it's going to cause that cell to then stimulate the release of FSH. So you'll still have that blood pathway and it will still bind to a cell. But you won't have a change in like ions. It just binds to that receptor and then it's like, okay, we need to release FSH. And then that cell will release FSH. It will go to the blood and all that. Did that answer that question a little bit?
Yeah.
Very cool. Okay. Quick secondary messengers. GPCRs is this first one. Super important. If you're pre-med, pre-PA, pre-vet, pre-nursing, whatever it is, you'll have other classes that talk about GPCRs in great length. So I think they're very important. So knowing, and I'm making it very basic, but essentially hormone binds, and we're going to talk about it in the terms of a hormone that actually stimulates a response. You can have some hormones that cause inhibition to the cell where things won't happen, but because we need to talk about what happens, we'll talk about one that stimulates it. So hormone binds, this is going to change the shape of this G protein, or this transmembrane protein. This is going to cause that G protein that I have in orange to come on over here. And it's going to interact with this transmembrane protein that somehow is going to cause a change in energy. So this G protein that's inactive is going to have something that we call GDP on it, guanine diphosphate. When it interacts with this transmembrane protein, it's going to be activated. And it's going to be given energy. So now it's going to have a GTP, so guanine triphosphate. So we add a phosphate to it. From there, this is going to make its way on over to another transmembrane protein called adenylyl cyclase. And adenylyl cyclase it takes ATP and it makes CAMP, which is cyclic AMP. And it's using the energy from GTP to do that. So you have to activate that G protein, get that GTP that has energy to then come over to adenylyl cyclase and actually activate that system to get to your CAMP. And then CAMP plays a bunch of roles. It's going to act on what we call PKA, or protein kinase A. Protein kinase A can then go off and increase ion channel activity. And it does that by phosphorylating it, or putting phosphates on the channel. That will increase activity. And then the other thing it will do is it will increase enzyme activity. So that's one of your first messengers, is that camp. Camp is very important. When we get to the heart, you're going to talk a shit ton about camp, and it's all very incredible, but camp is great.
So the PKA is specific to this example, not every time that camp is used.
To my knowledge, PKA is used every time with camp.
Okay.
Or it's some sort of kinase, protein kinase. I always learned it as protein kinase A because that's the one that will phosphorylate channels. And usually camp will cause some sort of enzyme to phosphorylate stuff, because camp itself is just a type of energy. So it will help to get those channels moving, or get those that enzyme moving and utilizing those channels and increasing more activity. So kind of the other one that kind of goes along with this, because it's the same GPCR situation. I just went ahead and skipped drawing out the whole transmembrane part. Let's just say all of that already happened. So the hormone bound, the G protein came over, got activated, has GTP now. This can then cause another cascade where we get calcium released from the ER. We can increase calcium activity through a channel. This can also, when that calcium enters, it binds to what we call calmodulin. And calmodulin is the one that can increase ion activity, or ion channel. But it also will increase enzyme. Or I'm sorry, this one does not do ion channel. That was my fault. This one just does it increases enzyme activity. I have this, 1 second. I'm trying to make this a little cleaner. Okay. So that calcium released from the ER will cause calmodulin. This is kind of a more fluffy chart, but when this is active, instead of doing the PKA camp route, you'll still have PKA involved, but you're not going through with camp. That PKA will phosphorylate that channel still, causing that calcium to come through. And we increase calcium concentrations coming from both the ER, or the endoplasmic reticulum, and from that channel. That will bind to that calmodulin. And then kind of the end all be all of this system is to increase enzyme activity. That's why we call that calmodulin our secondary messenger, because it's really what's going to increase whatever the enzymatic activity that you're trying to get to is within that cell. I think this one is the more complicated one. GPCRs to me feel a little bit less complicated. Obviously, there's a lot more that goes down in this. There's something with like IP3 and all these other things. I wouldn't stress too much about that stuff. I think just knowing that, really understanding that calmodulin and really what are the end all be alls of secondary messengers is that we're increasing ion channel activity and we're also increasing enzyme activity. And again, you can have inhibitory systems where you could have a G alpha, because remember the G protein is 3 parts, 3 subunits. It has an alpha, beta, and a gamma. Usually we're talking about G alpha S, that's a stimulatory, but we could also have a G alpha I, and the I stands for inhibitory. So that could actually inhibit the whole system. But do we have any questions on this before we go to the intracellular part? This stuff can be a little bit confusing, but I think the more you practice GPCRs and stuff, you'll kind of understand them.
So is it due to this effect on the ion channels why these are so useful in the autonomic nervous system with the catecholamines?
Yep. And like I like to the heart just makes so much more sense to me when it comes to this stuff, because you'll have epinephrine binds And you've talked about the ANS, so I guess I can kind of talk about it and it won't feel so foreign. But you have epinephrine or norepinephrine will bind to one of the heart muscle cells, and it will actually increase CAMP activity. And CAMP will actually bind to specific channels, and PKA will phosphorylate these channels that will cause more sodium into the cardiac muscle. And I know you're kind of getting into muscle contraction right now, but the more that you increase that positive charge in that muscle, it's going to cause contraction. So if you're running from a bear, because that's like the very typical fight or flight example we use, you're going to want more CAMP, more PKA, so that those channels can have more ions moving through. That's kind of the example I like to use, but that help a little bit.
Yeah, so this happens really, really fast.
Oh yeah. Like all of these processes sound like they take a long time, but no, they're like instant. Because we've all had moments where you are probably just sitting there and something scares you and your heart instantly starts beating faster. Like that's the autonomic fight or flight system working really quick. Cool. If we feel good about that, I want to keep moving so I can try to get to as much neuro. Okay, so now talking about secondary messengers when it comes to hydrophobic hormones, so our steroid, our lipid derivatives, cholesterol derivatives, all those things. So these will puncture directly through the cell, and it's going to bind to a cytoplasmic receptor or intracellular receptor, which is this orange guy or green guy here. So intracellular receptor. And I kind of enlarged it here just to draw this out. But these are going to have 4 different domains. The first domain is what we call our hormone binding domain. Makes so much sense. It's where the hormone's going to bind. Our second one is going to be our nuclear binding domain. So that is going to allow for this whole thing to come on over through the nuclear pore and enter our nucleus. That next domain is our DNA. I did not give myself enough room. DNA binding domain. This is what's going to let you actually bind to the DNA. So we have some DNA in here. It will allow it to bind there. It will read that DNA to figure out what part of the DNA we want to translate or transcribe and then translate to get the proteins that we need. And then our last one is going to be that what is it? Transactivation. That's what it is. Transactivation domain. So activating transcription because we want to make our mRNA. Once we have our mRNA, we can leave the nucleus. We can do our translation, and then we can have whatever the target response is. And the example I used earlier with aldosterone, that one wants to actually increase a certain number of transmembrane proteins that we have in the cell plasma membrane. So you'll actually increase translation of that channel and then put it onto that cell membrane, and that's what causes that sodium to be reabsorbed. But that's just a good example of how this system specifically works. And then it's really targeting genes themselves. Any questions on that? I don't know how far in depth he actually went with the domains, so I just went ahead and did it, but to have it make more sense. But that's kind of all the very brief stuff for endocrine. I'm going to now jump into like the actual hormones and stuff, but do we have any questions on just the brief endocrine stuff or the endocrine overview stuff?
Is the transactivation domain going to bind to the transcriptase itself?
I actually don't know specifically where it binds. I know that it acts as a transcription factor. So it's going to bind with all those other transcription factors that you have, which there's like a bunch of them. So I know that's kind of how it binds, is as a transcription factor. I don't know exactly where it binds though. It's a good question. Sorry, I can't answer it. Cool. Well, if not, let's jump into our hormones then. So I just drew this pituitary gland out, just so we can kind of go through the blood flow really quick. So we have our hypothalamus up here. We obviously have an anterior pituitary and then a posterior pituitary. Both of these will have some sort of blood supply running through them, which is how we get the hormones from the pituitary gland or the hypothalamus into the bloodstream themselves. I think it's really important to note where the posterior hormones are actually synthesized and where the anterior ones are. So starting with the hypothalamus, let's just do anterior first. That's one we're going to talk about the most, but let's just use GnR, or no, let's use, I don't know, thyroid releasing hormone as an example. I'm back. Do you guys hear me? Okay. I don't know why my internet just like dropped for a second. My bad. Okay, we're back. Here we go. Oh, they're recording. Oh, it says it's still recording. Should I? I don't know if I want to hit stop and then it like. Okay, I'm gonna pray that it's good. That's what I'm gonna hope. I think it's okay. As long as we all feel like it stopped and restarted. I see it like the red buttons on my screen and it shows stop. It says pause or stop recording at the bottom. So I think we're good. You were all here to witness that it wasn't me though that ruined the recording if it did ruin. It's my internet. Okay. I don't know why it did that, but let's keep going. So thyroid releasing hormone comes from the hypothalamus. It's going to enter the bloodstream. So I should say it's synthesized in cells in the hypothalamus. It will enter that portal system, and then it's going to make its way down to the anterior pituitary where it will leave the blood, and it's going to go to a cell here. It will bind to a receptor, cause some sort of change, and then cause the secretion of our thyroid stimulating hormone. This will then enter the blood again, and then now it can leave the anterior pituitary and then go to the target organ, which in this case is our thyroid hormone. So that's kind of how the anterior works. Again, we're going to talk a lot about all of those because we know that we need to know our flat pig abbreviation or mnemonic. Now, the posterior is kind of the more interesting one. It's more of a neural. So things are coming from actual neurons that are in the hypothalamus that extend down into the posterior pituitary and directly communicate with the bloodstream. What are the two major hormones that come from the posterior pituitary?
Oxytocin and antidiuretic.
I love that. Oxytocin and then antidiuretic hormone. Another name for this one, because I tend to use the other one a lot, just so you don't get confused, is vasopressin or vasopressin. Just so you know, they're the same thing. I learned it as vasopressin, so I totally spelled that wrong. Vasopressin, but just so you know. Okay. Where do you think these hormones are synthesized then?
Isn't it in the hypothalamus?
Yeah, perfect. Great. I just want you to know that, like, because with the anterior pituitary, we can get very caught up with, like, we know that certain hormones come from there. And yes, oxytocin and antidiuretic hormone are coming from the posterior pituitary, but they're synthesized in that hypothalamus, right? They're being sent down through a neuron. So just knowing that that's where synthesis is occurring is a good thing to know. Okay, let's do the dreaded 3-box model for all of these. So I had to kind of usually I would go straight across, but because I had to give you guys a PDF that went down, I had to like make it look a little bit weird. So I only wrote out the 3 parts here, but then when we go down, just know that each arrow indicates a different section. So we're always going to start with hypothalamus, anterior pituitary, and then the target organ, and then a target hormone. And then we'll go through the pathologies of each. So starting with which one acts on the testes and the ovaries. So I left the target organ there so we could at least have somewhere to start. But I'm going to make you guys answer these because I need you to know them.
That would be gonadotropin releasing hormone, right?
Yep. Yeah. So GnRH. And I know some people that came to my office hours know my little trick that if you feel like you forget where the hormone came from, if it has the term releasing hormone in it, so like gonadotropin releasing hormone, I think there was another one, thyroid releasing hormone. Releasing tells you it's coming from the hypothalamus. So if you see releasing in the name, you know it's the hypothalamus. That's just a little trick if you're like, shit, I totally forgot where that came from. Cool. What are the 2 hormones that this causes to be released from the anterior pituitary?
Follicle stimulating hormone and luteinizing hormone.
Perfect. Yeah. And both of these actually act on testes and ovaries, depending on what you have. And they'll cause certain target hormones, specifically androgens, which are basically testosterone, steroid hormones. Another one is called inhibin. You guys are going to talk about this system a lot more in depth when we talk about reproduction, but inhibin is one that actually negatively feedbacks to the loop. But I typically just go with androgens, estrogen, testosterone. Those are kind of the things. And he did not talk about really any pathologies with this system, so we're not going to worry about it. Let's move on.
He did talk about precocious puberty, I believe.
Oh, did he?
Precocious puberty and hirsutism.
Oh, the hair one. He did talk about that.
Yeah, the hair one, that's it.
Oh, yes, the hair one. Okay, so we have the puberty. I don't even know how to spell that.
Precocious.
Perfect.
And then puberty.
Cool. And then the other one was the
Hirsutism.
I won't be able to spell that either. Is it H-I-R?
Hirsutism. Hair. Lots and lots of it.
Yeah, that one is, and I actually don't really fully, did he talk about the reason why that happens? I don't remember.
Yeah. So an
Oh, yeah,
excess of one of the hormones, I want to say it's FSH,
yes,
causes an increase in testosterone and androgens in people with ovaries. And this causes excess hair growth on the face, namely, due to this increased testosterone. Ish, thereabouts.
Okay, yep. That's all I would know about it.
Play.
Yeah, because this is like, it's a gonadotropin. It's if you have like a tumor located in like the hypothalamus area. Because you're going to increase, kind of like the general rule of thumb too with systems like this is if we were to ask you a question about a tumor being somewhere, so if we said there was a tumor in the hypothalamus, then whatever hormone the hypothalamus is actually producing is going to be at super high levels. And it's going to cause all the hormones that are under it to also be very high levels. So in this case, we're talking about a tumor that's causing gonadotropin releasing hormone to have an issue, which then is going to increase everything under it. Because if we're making more gonadotropin releasing hormone, then we're going to make more FSH and LH. And then that's what's causing that over excessive hair growth. Cool. So pathologies for those. I think that's pretty good on that one. What about the adrenal glands? So what are these hormones? This is the HPA axis. So hypothalamic pituitary adrenal gland axis.
Is one of them, aren't there, there's a couple, right?
So think back to which one usually comes from the hypothalamus. So starting from the hypothalamus down.
Is this going to be antidiuretic? Diuretic, if I can English.
So antidiuretic comes from what part of the pituitary?
Oh, pituitary. I lied.
No, you're
Wait.
good. It
No,
comes
that's going to
from
be the
pituitary.
posterior pituitary,
Exactly.
which comes from the hypothalamus,
Mhm.
right?
Yep.
Which will then act on the kidneys, hence the adrenal gland.
Yes.
Yay.
I know they can get very with how many hormones you have to know, they can get a little bit.
There's so many.
So many. So this one's gonna be our corticotropin releasing. Remember, releasing hormone is that big. I would write it all out, but I just don't have time. Corticotropin releasing hormone comes from the hypothalamus. This is going to act on the anterior pituitary, and it's going to release ACTH, which is adrenocorticotropin releasing, adrenocorticotropin hormone. That goes to the adrenal gland, and it kind of tells you adrenocorticotropin. Adrena could just tell you adrenal gland, so it's going to act directly on that. And here we're going to end up with our cortisol, or like our stress hormones, or what we could also broadly call our glucocorticoids. Now, here's something you guys definitely do need to know. What layer of the adrenal cortex do the glucocorticoids come from?
Glomerulosa.
Are you sure?
Fasciculata.
I'm pretty sure it comes from the exterior.
Fasciculata.
Fast. Oh, hi, Kitty.
It's the middle layer? Okay.
Yeah. So,
So-
remember salt, sugar, sex.
What's really nice is if you can know, like, essentially the layers he talked about all make up the cortex. And when we talk about the adrenal gland, it has a shape, like a weird bean looking shape, sits on top of the kidney. The whole inside is what we call the medulla. And the medulla only makes two things, epinephrine and norepinephrine, or our adrenaline hormones. Everything on the outside, so all of these layers are considered the cortex if we were talking about them as a whole. So when I say adrenal cortex, that's just talking as a whole. If we're being really specific, then yeah, we would say it's the F one. Because I don't even want to try to say it. Adrenal. Or whoops, zona. Sorry. So it's coming from the zona. Fasciculata. But what are some of the pathologies with this one?
Diabetes insipidus.
Yeah. What are some of the other big ones? Where did I write them here? Pathologies.
Is this Cushing's disease and Addison's disease?
Those I think are going to be the diabetes one is important, but these two are like the really big ones that I think are good to know. So Cushing's, and what is Cushing's? What causes the Cushing's? Is it too much?
An overproduction?
Yeah, of what?
Cortisol?
Yep. So overproduction of cortisol. And then what about Addison's?
A decreased production in cortisol.
Yep. Addison's disease. Decrease. Cool. So yeah, I think kind of just knowing those pathologies, what goes along with them, is going to be important. Like I said, if I could go into more detail on them, I would, but I'd want to try to get to neuro. So look at the slides, but know Cushing's and Addison's. I think those are very important when it comes to this system. And then know your levels of the adrenal cortex. All right. What about thyroid gland? What's acting on the thyroid gland?
Thyroid releasing hormone.
Awesome. So thyroid releasing hormone, causes the release of which one?
Thyroid stimulating hormone.
Yes. Stimulating hormone. Cool. That acts on the thyroid gland. What is the end all be all hormone?
T3 and T4.
Which one's more potent?
T3.
Perfect. Cool. So that's kind of the one we're like really looking for there is that T3. What are some of these diseases or pathologies?
This is going to be that myxedema, congenital iodide deficiency syndrome, Graves' disease.
Yes, Graves' disease. The iodine, because iodine plays a huge part in that conversion of getting to T3 and T4. To be quite honest, I took pharmacology and we learned about this process, and we didn't even have to know that whole iodine swap and stuff that actually happens in the adrenal gland. So I wouldn't stress too much about the details in that system unless you're taking like a higher level class later on. But just knowing that T3 is kind of that more potent one. But that deficiency can cause some issues, obviously, because you need iodine in that system to get to T3 and T4. Cool. And next one, so mammary glands. Which one works on the mammary glands?
Prolactin.
Perfect. So we have prolactin. What comes from the hypothalamus? Which one specifically?
Some good old oxy right there.
So this will be oxytocin will help. Yes. What does the oxytocin one do? Is it milk letdown or milk release?
It's the oh wait. Prolactin creates milk. Oxytocin helps with the release, doesn't it?
Yeah. Yep. So, and usually oxytocin, I think, also plays a very large role in actual labor too. That's kind of the big one for the part.
Positive feedback.
Yep. It has a huge positive feedback. This one is going to release that prolactin. But this one has kind of another hormone called prolactin inhibitory hormone. Oh, whoops, I meant to draw an inhibitory. This will actually inhibit the release of prolactin. So the hypothalamus can either release prolactin releasing hormone or prolactin inhibitory hormone. It makes sense if you're not pregnant or you're not breastfeeding, you probably don't want to just be like producing milk all the time. So you're going to want some sort of inhibitory system going on. So this one kind of has another one that comes from the actual hypothalamus. And then I don't remember if he talked about any pathologies. So remind me if he did.
Galactorrhea. He very briefly touched upon that. That occurs when a prolactinoma is, stuck in your mammary glands or even in people without mammary glands. So think biological males. They will have a buildup of prolactin and their nipples will be leaking all the time, and that is not normal nor fun.
Yes. Okay, yeah, I see it. Cool. I'll write that one and then just kind of know the little deets on that guy. Even though I really think it's so briefly stated, I wouldn't stress too much about that one. What's it called?
Prolactinoma. So essentially just write down the hormone and then
some kind of cancer going on.
I think it's actually a benign
it's like
tumor.
growth.
Exactly, kind of like a cyst almost. At least I think.
At least they could probably remove those treatment.
Yeah, I hope so.
I hope so too. Okay, last big one that I think is actually going to be pretty important is our and I wrote liver. It kind of acts on a lot of epithelial tissues, but this is going to be our growth hormone. And actually I meant to add, so this is growth hormone releasing hormone. Coming from that, we get our growth hormone. That's going to act on the liver. We end up getting somatomendins. Somatomendins can actually play a pretty big role in stimulation. So, and actually they play more of a role in stimulation of another hormone called growth hormone inhibitory hormone. And they stimulate its release. So they cause that to be stimulated. So it's actually somatomendins are going to inhibit this system. And they actually play that other role. They inhibit the releasing part. So somatomins are supposed to kind of help if you have too much growth hormone going on. They're supposed to help start to stimulate the inhibitory hormone, so we kind of stop making it, and it actually inhibit the releasing hormone. So it kind of plays both roles there. But what are the two big ones for this guy?
Gigantism and acromegaly.
Well, which one is a deficiency?
Neither are deficiencies.
Good. They're all too much.
Yep, it's a matter of when in life you experience that onset. If you experience excess growth hormones before the fusion of your epiphyseal, whatever, those growth plates,
Oh yeah,
then you are,
plates.
yes, thank you.
Yeah.
You're likely to deal with gigantism because
Mhm.
your body's just pumping and pumping out these growth hormones, and your body can't really stop it because these plates aren't fused, right?
Yep.
So you're going to be heckin' tall. And if you have fused growth plates but excess growth hormones, then you will deal with acromegaly, which is going to deal in, or it's going to impact your extremities, some organs, and enlarge them, deal with a lot of other complications.
Yeah, so actually this one, if you had a tumor here later on in life, is usually what causes acromegaly. Usually, I mean, I shouldn't say it's not like all the time, but like I have a close family friend who had a tumor in his anterior pituitary, caused this to increase. So that caused this to increase. So then his ears, nose, feet, hands got like extremely large compared to the rest of his body because of that tumor. He got it removed and he's fine. But I mean, his ears and nose are still large and his hands are huge. But that's kind of just an example of like that tumor idea and how, and that's a later on in life thing. Usually you're not born with the tumor on the hypothalamus. But anywho, I think those are all of the hormones that I feel are necessary. And I have 30 minutes to try to do neuro. Do you guys have any questions on endocrine? I'm going to try my best. Cool. I didn't have notes on these just because it took me a long time to do the other ones. So we're just going to wing it. I'm going to go through what I feel like are the most important parts of neuro. I'm not going to do a bunch of little detail stuff. So let's really just start with organizing CNS, PNS, and talking more specifically about glial cells. What glial cells do we find in the CNS? So the supporting cells. Obviously, we find neurons everywhere, but
Astrocytes.
perfect.
Oligodendrocytes.
Yeah.
Um,
And we know
there's two more.
I know.
Two more. I have exactly half of a brain cell right now, so I couldn't tell you.
So one is ependymal.
Oh,
Yes.
duh.
Those are doing our CSF. And then the last one is going to be microglia, which we didn't talk much about, but they are essentially a defense mechanism. So they eat up bacteria and other things. In the PNS, we have what's the kind of like the astrocyte, but in the PNS?
Satellite.
Perfect. I'm just gonna spell it so wrong, whatever. Satellite, whatever I spell. And then what about for the oligodendrocytes? What's the one that myelinates in the PNS?
Schwann cells.
Bronze.
Yep, Schwann. Awesome. Cool. Know these distinctions. Know, like, if I were to ask you a question about oligodendrocytes, you should kind of just know like where you are in space. Are you in the CNS or the PNS? Knowing kind of where that's at is important. All right, let's draw a big neuron and go through that really quick. So I'm not going to break this down anatomy or structure of it because I feel like we should all really kind of understand that just from basic biology. But we know cell body, axon, axon terminal. These are my dendrites. What I want to break down is where we find certain channels. So what channels do we find in the input region here?
Ligand gated ion shell channels.
Nice. So ligand gated. What's the ligand we're usually talking about?
Great question.
Acetylcholine.
Yep, could be. Well, yeah, I guess I should have been more more specific or general with my question. Neurotransmitters in general, right? That's kind of the ligand we're thinking about. I know that was kind of my fault. But neurotransmitters are the ones that are actually, I should not write that there. Let me change that. Neurotransmitters are the ligand. And when the neurotransmitter binds, does the neurotransmitter go into the cell?
No, it triggers a graded potential,
Perfect.
which may open the voltage gated channels if it's big enough.
No, that's great. There is one more channel we're missing, though, that's in the input region. This channel can cause either membrane resistance to be low or high. And it's kind of located everywhere. Helps maintain gradient.
Leak channels?
Yep. So we have potassium leak. Remember with resistance, it's all about if you have more leak channels, you have less resistance. Or if you have more or less leak channels, you have more resistance, stuff like that. Cool. All right. Next one. So in the axon, what are our channels that are in the axon?
Voltage gated channel.
Awesome. Voltage gated sodium and potassium. And then what else? Some lovely potassium leak channels again. And then kind of in the axon terminal, we just have one specific one that I really want you to know.
The voltage gated, the calcium.
Yeah. Cool. So knowing just where that is at is really important. And I actually already liked that Gray was adding in about graded potentials versus action potentials. Where would we find a graded potential? What part of this neuron?
It's going to be in the soma slash, I forget what it's called, but that one region in between the soma and the axon in which the cell essentially decides, okay, what am I going to do with this potential?
Yep. So graded potentials are always in the input region. They can be variant, meaning they can change. They can either be, you could have a bunch of excitatory postsynaptic potentials that cause it to go up and be depolarized, or they can be hyperpolarizing. Whatever it is, they can variant. They can be different. They can move faster, move slower, depending on summation and where the signals are coming in temporal wise or spatial wise. They can be variant. Now on the axon part, we should all know it's just an action potential. So if we're able to get a signal from the input region that's high enough to hit threshold, we're going to have an action potential, and action potentials are invariant. They do not change duration. They do not change anything. They're an all or nothing. So if you hit threshold, it's going to happen. And we don't really have a potential that happens within the axon terminal. That action potential will just make its way down to then open up those channels at the end. There is another type of channel or potential that happens in the input region, but it's on specific types of neurons. What's another type of potential that you've talked about?
What'd you ask again?
Oh, there's another kind of potential that happens. It's called a generator. Please tell me if I say something that he didn't actually talk about.
We were told they were essentially one and the same. They could be interchangeable. That's at least what I gathered, though. So anyone else, pipe in and let me know.
So I mean, okay, I will say essentially they're the same in the way that they are in the input region and they have to summate to get to threshold to have an action potential. The way that they're different is graded potentials always occur at a first order neuron due to usually mechanically activated channels or stretch activated channels. So like me touching my finger opens up mechanically gated channels, so not ion channels, because I don't have like an ion on my finger or a neurotransmitter on my finger that's opening a channel, right? It's just like that stretch that opens those channels. That is where you'd have a generator potential because you don't have two neurons there, you have just the one. So at this I would write no post synaptic neuron. So you don't have a neuron that's communicating. It's just due to touch or temperature or whatever, you know, I always just say sensation because that's what we're feeling on our skin. And you know, your skin is not, or your hand is not a neuron. So it's some other type of potential. If he did
Do
say,
you mean presynaptic?
no, postsynaptic, because it would be post the synapse.
Okay.
Oh, no, wait. Maybe. No, wait. Presynaptic would be before the synapse. You're right.
Presynaptic
Yeah,
would mean there's no neuron before.
yes. Sorry. Presynaptic, no presynaptic neuron. So like if I were to draw, here's my lovely hand. First order neurons usually have their input region here. So here's my input. This is where you'd have your generator potential. This would summate right here. And these neurons are different because, and I know we'll get to it, or I'll try to get to it, but it's going to be where they have a dorsal. They have their cell bodies like in the middle of the neurons. They have two. It's a unipolar. They have just the one in the middle. So here's an input region, and then our output would be in the spinal cord. That generator potential, once it hits threshold at this point right here, then that action potential will run all the way to the output in the spinal cord, and then send that signal wherever it's going. But that's kind of where I was getting at with generator. But if he said that they're essentially the same, then I would just go with what he said. But I would just wanted to clear that up a little bit. Okay. Now that we have this, let's go action potential graph. It's like very important to know. So value-wise, -70 is our resting membrane. What's the equilibrium potential for potassium?
-98.
Cool. Sorry, he gave you different values than I know, so I'm asking you guys. I always knew it as -80. I think they could be different values all the time. I think know the ones he gave you though. So -98 is our equilibrium potential for potassium. What is our equilibrium potential for sodium?
I think it's -65.
For sodium?
I'm sorry, +60. Sorry.
Oh, okay.
Chloride is -65.
Yes. So positive, wait, 60 he gave you?
+60 for
Okay.
the equilibrium potential.
Yep, and then -65 is the one for chloride. So we know, well then that kind of just, I put it under there for some reason. Okay, -65. So that's for chloride. So EQ of chloride. And then our EQ of sodium is up here. And the reason I like these values is they tell you, depending on what's moving at the time, the dominant ion that's moving is the equilibrium potential that we're going to seek. So if sodium's the ion that's moving the most, because we know we have leak channels still, then we're going to try to get to that +60. That's why the graph kind of does what it does. So starting off at resting, let's just say hypothetically, we start getting some summation of EPSPs. So what type of ligand gated channel is opening for EPSPs? Or for what ion? An EPSP is excitatory. IPSP is inhibitory. So if we're getting summation of EPSPs, so we're getting excitatory, what is the ion that's entering the input region?
Would it be sodium?
Yes. So sodium. Let's say we just get like one little EPSP. And we bump up a little bit. We're not, this is our graded potential, right? We haven't hit threshold, so we don't have an action potential. But then let's say suddenly we have an IPSP come in. What ions come in now? Inhibitory postsynaptic potential. And we would actually dip down back to resting because it basically they kind of equal each other out. But this would be due to chloride coming in, right? Even though I don't like that he has chloride. We'll go up there. But if we have a few of these, we would stay at chlorides negative, right? So if we have some IPSPs coming in and we're like, okay, well, it's negative 65 and thresholds at negative 55, we can't have an action potential. But then let's say some more stimulus comes in later, and then we can actually grate all the way up to there. This is kind of, I'm just trying to demonstrate that within a neuron, you can have IPSPs and EPSPs coming in, and they can cause these summations where your graded potential looks very different. And it really only happens once you hit threshold, then you get that action potential. So it's really depending on what the stimulus is. But once we hit threshold, we know that our equilibrium potential for sodium is positive 60. So that's going to jump all the way up there. And then we have to repolarize, but we hyperpolarize because we have to get to the negative or the equilibrium potential for potassium. And then it's going to come back up to resting. So I'm going to label this really fast and we'll try to go through it quickly. So what's happening at 1?
Depolarization.
Yep. What channels are open at depolarization?
Potassium.
So we do have potassium leak, but what's depolarizing that membrane?
Sodium.
Oh.
And it's that voltage gated, right? So I will be honest, if it's a fill in the blank, this is just where I want you to really know your channels. Don't just put sodium channel, right? Because there's different types of sodium channels. There's a ligand gated, so it opens due to a ligand. There's a voltage gated. So just know, like, make sure you put what type of sodium channel it is. So in this case, it's voltage gated. At this point up here, which I'll just put as 1.5, is where sodium channel or voltage gated sodium channels inactivate. So they don't close, they're just now in
a ton. It is the best water park I've ever been to.
Now I want to know what water park. What water park is the best water park? They're gonna kill me for even calling them out on that. I'm so sorry. Okay. 3 is repolarization. Which is going to be due to voltage gated potassium. That's why we dip all the way back down to negative 98, which is our equilibrium potential for potassium. And then all of our channels close here. So all voltage gated close. So now we can then repolarize back to our, or depolarize back to our resting membrane potential, which is at negative 70. And that's all due to our potassium leak. And we have sodium, potassium, ATPases, which are channels that utilize energy to help swap sodium and potassium. So I think it's like 3 sodium out, 2 potassium in, or is it the other way around? I forget. My brain's going blank. Anyway, know this
You're
chart.
right, because it's more positive out than positive in.
Okay, yeah. Yep, so that's the way those are moving, but that helps maintain our resting membrane potential at that -70. Oh shit. It didn't die, it just does this. You guys have any questions on that? I would say totally no. That or that, what am I trying to say? That action potential graph really well. Just know your parts of it. Look at that lovely picture of me in the corner. Great. Okay. Cool. What else do I need to do for neuro? There's pathways. I have 10 minutes. What do you guys want to know really quick about neuro? What do you feel like you need to go over?
He said we absolutely need to know that pathology where you have damage to just one side of the spinal cord and how it causes like loss of motor and then loss of
Yes.
fine touch.
You like
That's on each side.
180,000% should know that. Okay, let's do it really quick. Actually, the pathways are literally my favorite part of this whole unit. So you guys like neuro? They have a whole, I think it's neuroanatomy, but they do every pathway, which I think would be pretty sick. Okay. So if we're talking about, so this is our right and left. Our dorsal column, or he calls it, sorry, I call it dorsal, even though it is not ventral, sorry, posterior. Posterior medial lemniscus. Posterior column medial lemniscus tract. So posterior sits in the back of the spinal cord. Here's my lovely spinal cord. I'm not going to label it, but this is going to be our ventral, our anterior horns and our posterior horns. I always learned it as dorsal ventral, which basically mean the same thing. Dorsal's back, ventral's forward. They use that for animal anatomy, but for people we use anterior, posterior. So if I say dorsal, just know I'm trying to say posterior. I just learned it dorsal. That's why I'm so confused. Okay, so posterior, anterior. We know this is white matter. This is gray matter. Here's our central canal. So if we're talking about the PCML, which is the posterior column medial lemniscus tract, our first order neuron could be all the way out here in a finger. It's going to come in and it's going to directly enter, or where does it leave its cell body? Leaves it in our posterior root ganglion. So we have a ganglion, which is a cluster of cell bodies in the PNS. So not in the central nervous system, but in the peripheral nervous system. And it's going to leave those cell bodies there. From here, this is going to ascend up this dorsal part over here, or the posterior part in the back. So it's going to come up the spinal cord, and it's going to synapse onto our second order neuron, where it crosses the midline. Second order neurons always cross the midline. That's a rule. So they always will cross. This happens in our posterior. I'm trying to remember what it's called on the dorsal. Nuclei. It's a nuclei. So nuclei, where it's another name for a cluster of cell bodies in the CNS is a nuclei. So that's where you have a cluster of cell bodies. This is then going to cross the midline. So I've come from the right side. I'm now on the left side since I've crossed that midline. And then I'm going to have the rest of my third order neuron, which synapses in the thalamic relay nuclei, which is in the thalamus. It's like a bunch of, it's right in the center of the brain where all these neurons cross paths or synapse onto each other. And then I'm going to talk to my somatosensory cortex, where then I can have some sort of motor response. So that's our PCML. What are the modalities that the PCML bring? It's like, what are we sensing with the PCML? So it's going to be fine touch. I think this is the vibrations one. And then the big one is proprioception, which is where we are in space. So knowing like where my hand is in space is due to this tract. Cool. Next one, spinothalamic. We'll draw another first order neuron out here. Leaves its cell body in the posterior root ganglion. This one is going to enter the posterior horn, but it's going to synapse directly right away onto our second order neuron. Second order neuron crosses the midline. It's going to enter the spinal thalamic tract, which is right here. And it's going to ascend. And it's actually going to go to the same place, thalamic relay nuclei. And then it's going to have its third order neuron synapse in the somatosensory cortex. The SST, so spinal thalamic tract, carries pain and pressure. And I'm only going to draw the one side, but let's say I had a lesion, and usually the questions we like to ask are if it's halfway. If we completely severed the spinal cord, anything under where we severed it, everything's lost, right? So if it happened right above the legs, then you would lose everything to the legs. You're essentially paraplegic. So the idea, paraplegic. The idea is that if we were to put a lesion right here on the right side, so let's say you had a cut, or let's say it's a tumor or some kind of growth. So you have a lesion here. What would we lose? What type of sensation would we lose?
Fine touch, vibrations.
On what side?
Oh, on the right side, but then you would lose pain on the left side.
Beautiful. Yep. So what I highly recommend you do, if you have a question on this on the exam, is draw the pathways right next to it. Put where he said the lesion was, and then go through which ones you're losing. Like, just draw the pathways really fast. It can be like the crappiest drawing ever, but like as long as you have it right next to you, then I think it's the best way to really realize, okay, this is what is actually cut, right? And if we were talking about motor, so motor, let's say this is the motor cortex. We have a long ass motor neuron that crosses the midline up here, and then it comes directly to this side where it synapses onto a lower motor neuron here, goes out to the muscle. So if we had that lesion there, what side are we losing motor on? So same lesion.
The right?
Yep. Yeah. What's nice is with motion, whatever side the lesion's on is where you're losing motor, because it crosses in the brain. It doesn't cross down in the spinal cord. So that's that upper motor neuron always crosses up in what we call the internal capsule, which is like in the brain area. I don't actually know exactly where it's at. I think it's kind of by the thalamus, but it crosses over there and then comes down. So whatever side the lesion's on is where you're losing motor. Yeah, I went through that really fast, but do you guys have any other questions? I'm sorry I couldn't get through all of neuro. Please ask away. I have online office hours tomorrow. I think I'm going to push the time back just a little bit, like an hour. But if you feel like you have any other questions, it's online. You can stop by on Zoom and just ask away, and I'll get through more neuro stuff tomorrow for that. But also just go with confidence if you feel great. Also, don't.
So it's the third order neuron that ends in the thalamus.
Third order neuron is where the cell body is, or the cell body for the third order neuron is in the thalamus, and then it actually ends in the somatosensory cortex. I didn't label that. So the somatosensory cortex is part of the cerebral cortex, so it's on the outer part of the brain. It's the part that kind of surrounds the inside part. Yeah, thank you guys. email to, if you have any questions, you can email us too. We've been trying to get back as quick as we can. And hopefully this teaching style helped kind of jog your brain on getting through some of the stuff.
This was wonderful. This is exactly how I learned, so I loved it.
Good. Okay, good. I know I always am iffy if I should draw because I feel like it takes way more time, but I think you get
So
more.
worth it. It looks so nice.
I know I'm
Yeah,
so
it makes a lot of
with
sense. I like it a lot.
my lovely bone drawing, and I was like, well, I don't have any neuro stuff, so here I go.
That bone drawing was a work of art.
I know I really spent too much time on that.
Well, it looks great and it was very
Yes.
helpful. So thank you.
Yeah, of course. Thank you guys.
Thank you.
Yeah, good luck on the
Bye.
exam. I'm gonna end the recording now.